Subject right now is zebrafish and what they can tell us about brain synapses. These are zebrafish, and here is the male and on the right is the female. They're actually really useful in the lab, partly because they reproduce quickly. They also are very, very easy to keep alive. And even though it might not be obvious with these particular photos, it's not really hard to tell the males and females apart. You can see the female is kind of bigger in the abdomen and the male is darker in the stripes. But what really makes them useful is their development, particularly their development up to about 10 days or so. Because you can see, you got all the egg and how it's developing. It's all transparent, and in fact, after they hatch, they continue to be transparent all the way up through at least day 9. This is the action of a molecule called FingR, F-I-N-G-R. I tried to find a picture of the actual molecule itself, but I couldn't find one. But what FingR does is it sticks to different proteins in brain synapses. So synapses are where the neurons connect to each other. And at those points of connection, basically those neurons send chemicals back and forth, neurotransmitters back and forth, and that's how basically all the communication in our nerves happens, in our brains, our spinal cord, all of that stuff. So this is the study I'm going to be talking about. Sleep pressure modulates single neuron synapse number in zebrafish. And obviously, even though this is about zebrafish, the assumption is that it has some application to human beings. And honestly, there's no reason to think it wouldn't at least be similar. So there's a few different ways to get this FingR protein into the brain of the creature you want to look at. One way is get it into the parents, and then the parents can pass it on to the offspring. And then because these zebrafish are transparent, you can just shine a UV light on them and you can see the different synapses. Of course, it's really small, so you got to get close in there with probably with a fiber optic camera to be able to see the actual synapses themselves. There was another study done where they took mice, and what they did, they put the FingR protein RNA, the RNA to produce it, into a viral capsule. And those little viral capsules went and infected the mice's brain. They didn't replicate in there, but they did cause the mice to start producing FingR in all their neurons. And then you could see all the different little synapses. Of course, in order to see the synapses, they had to drill a hole in their brain. Or I'm sorry, drill a hole in their skull. In this particular study, they basically used an mRNA kind of vaccine and just injected that directly into the brains of the zebrafish larvae. And then they were able to use that to light up their neurons. The bulb with the blue arrows, that's actually the nucleus of the neurons. It's the main body of the neurons. And these are all the little dendrites and axons that come off of it. And then you can see on these little white parts, so the membrane is the magenta, and then you can see on these little white parts, that's actually where the synapses are. That's where they're connecting to other neurons. And then once they could see those synapses, they could give them different IDs and identify them by different locations on the neurons. So they knew which specific synapses were being affected by their experiments. So the question was, what happens during sleep with these zebrafish? What do the synapses do? So in our brains, obviously, there's neurons and microglia and astrocytes, all these different types of cells. Specifically, they were just looking at the connections between different nerve cells. But while we're awake, the number of synapses, the number of connections increases. And then while you're asleep, the number of connections decreases. And what that is, that's actually got a lot to do with these microglia, which are kind of custodians for neurons. They kind of take care of them, but they also clip out some of the synapses that don't seem as useful. So in order to look at some of these dynamics during sleep, they basically used paintbrushes to keep some of these zebrafish awake. Now the zebrafish were sleeping for, it looks like, about 10 hours a day. And so they basically had probably some poor, unfortunate grad student sitting there for 4 hours keeping zebrafish awake with a paintbrush. But the ones that were allowed to sleep, those are in the blue. And you can see in the first half during sleep, the number of synapses they had every hour declined by about 1 to 2 synapses every hour. And then during the second half of their sleep cycle, the number of synapses slightly increased. So during that full night of sleep, during that full cycle of sleep, there was pruning and then there was regeneration. So it's like a complete remodeling of the things those zebrafish had learned during the day. Well, if you take the ones that were sleep deprived, they continued creating synapses that whole time that they were sleep deprived while they were kept awake, which is kind of what you would expect. But then when they were allowed to sleep, there was a very swift pruning of those synapses. Now, what you'll notice is there isn't another phase there. There's just that pruning phase. There was no adding back on. So there wasn't a full remodeling of memory during their sleep cycle. And what this indicates to me is that when you're sleep deprived, you lose a lot of your ability to learn. Now, one thing that's interesting is that the early sleepers, during their synapse loss phase, they lost more than the ones that were sleep deprived. And what that might indicate is either that there was less pruning that happened, or their brain was actually trying to clip out and build on all at the same time. So then what the researchers did was tried to look for kind of a molecular basis for this, or at least a neurotransmitter basis. And specifically, the two neurotransmitters they looked at were adenosine and norepinephrine. So adenosine receptors are found throughout the brain. You can see there are different types, and different types can show up on any one cell. And this shows which different types are in different segments of the brain. But during the day, adenosine kind of builds up and builds up until you get so much that you feel like you must sleep. And then while you're asleep, that adenosine is broken down. So that kind of allows you to wake back up. The second neurotransmitter they looked at was norepinephrine. Norepinephrine is not just produced in the brain, obviously. It's produced in the adrenal glands also, where it kind of operates as a hormone. But norepinephrine pushes the brain to stay awake. So you have this push and pull between adenosine, which wants you to calm down, and norepinephrine, which kind of amps you back up. And you can see it's pretty much produced in this locus ceruleans and pumped out through the rest of the brain. It's actually produced from a modification of dopamine. But whether you're talking about adenosine or norepinephrine, if you get enough adenosine in you, you will kind of wink out. And if you don't have enough norepinephrine, again, you will kind of start to fall asleep. So they gave these fish a couple of different drugs. One is clonidine, which kind of mitigates or suppresses norepinephrine, and then chloroadenosine, which amps up the action of adenosine. And they looked at how it affected their sleep. And each of them by themselves did put the fish to sleep. When they put them together, the fish really slept. Right here, this shows the synapses, and the green is the control group. So you can see the green group didn't really lose synapses. It kept gaining them. And when they gave each of the drugs by themselves, they still continued to gain synapses even as they slept. But the group that they gave both to actually lost synapses. So rising adenosine and falling norepinephrine are both essential for remodeling memory.